Protavio is very important in the DIOPTRA project. Our goal is to discover a set of protein biomarkers that are present in blood that can be used to differentiate between the DIOPTRA study groups or the study populations that we have in DIOPTRA, which are people that are healthy, people that have non-neoplastic findings in the colonoscopy, and people that have advanced neoplastic findings, and the final group is the group that has colorectal cancer. In order to identify and discover these novel protein biomarkers, we are analyzing matched samples from DIOPTRA participants, matched being that we are analyzing both tissue samples and blood samples from the same participant. The blood samples are collected prior to the colonoscopy, and the tissue samples are collected during the colonoscopy in the samples that the gastroenterologist finds during the procedure. And for the blood samples, we are doing a proteomics analysis, and for the tissue samples, we are doing a transcriptomics analysis. So this biomarker discovery pipeline that we are developing, which is very novel and state-of-the-art, has what we call a multi-omics approach, because we combine both proteomics from the blood samples and transcriptomics from the tissue samples. We have almost finished with the sample collection, set up all the instrumentation for the pipeline, which is both for the proteomics and the transcriptomics. We have developed all the protocols and validated most of the protocols for some samples that we have available so far, and we have optimized everything. So the final thing now is to collect all the samples and do the final experimental design, where we'll do all the proteomics analysis and transcriptomics according to the pipelines that we have optimized. And the important part of this biomarker discovery pipeline is that it is very novel because it uses state-of-the-art technologies like Olink proteomics and RNA sequencing. And for Olink proteomics so far, we have received a certification from the provider of the Olink methodology. So far, we have organized the first pilot experiment, which is a feasibility study of this whole pipeline, where we will analyze 88 samples, but not with the full potential of the biomarker discovery pipeline. So our goal is to validate all the processes that we have developed so far with these 88 samples to see that everything works correctly. And then we'll do the design of the final discovery experiment by September, and we will have the full discovery data set by the end of the year, along with the analysis.